Welcome to the Ohio Attorney General's Center for the Future of Forensic Science Lab on Bowling Green State University's campus. Here at BGSU, we offer a Bachelor's of Science in Forensic Science with a specialization in chemistry, biology, or examination, and a Master's of Science in Forensic Science with concentration in biology and chemistry. If you choose to participate in our accelerated program, both can be earned in just 5 years. Our $2.1 million Forensic Science Lab was opened in early 2019. This world-class lab is designed to mirror a real operating crime lab like the branch of the Ohio Bureau of Investigation that's located just across the street. Because of this, forensic science students here have the opportunity to learn from professors with years of forensic science experience on some of the most current state-of-the-art instruments to prepare them for a successful career in forensic science. In our lab, we have 2 comparison microscopes. The comparison microscope in the lab area is comprised of 2 compound microscopes connected by an optical bridge. The bridge allows the user to view samples on each microscope simultaneously. In TRACE, we use it for visual microscopic comparison of hairs and fibers. The firearms comparison scope is the same dual microscope with an optical bridge design, but the working distances are configured to accommodate larger samples. The scope is common to crime lab firearm units. It allows for a direct visual comparison of recovered bullets and cartridge cases with known test-fired specimens. The lab also features an entire wall of compound and dissecting scopes for student use in the classrooms. These are used quite extensively in many of the classes for everything from visualizing trace evidence to biological sample analysis. This is our FTIR. It is a Fourier transform infrared spectrophotometer. It's equipped with a microscopic visual component that allows for analysis of small samples or specific areas within samples. In the trace evidence unit, we use it primarily to analyze, identify, and compare chemical constituents of certain types of polymer-based evidence. This includes paint, synthetic fibers, assorted plastics, and adhesives. Here is our ductless fume hood from Air Science. This hood is typically used for manual organic DNA extractions. This is the superglue cabinet. It is also a ductless model from Air Science. This cabinet is used in fingerprinting. Cyanoacrylate or superglue fuming uses the vapors of superglue to develop or make latent prints visible. Superglue fuming can be performed on old or new fingerprints and can then be further enhanced by using dyes or powders. Our lab also features a GCMS instrument. GCMS stands for Gas Chromatograph Mass Spectrometer and is used to separate and identify components of chemical mixtures. The instrument separates the components of a mixture with the chromatography portion and then identifies them with the spectrometer portion. This is used in the crime lab to identify drug samples and unknown powders. This large instrument is our Shimadzu LCMS. It is similar to the GCMS but uses liquid chromatography instead of gas chromatography. This instrument is used to separate and identify components of chemical mixtures but can also quantify those components. It is mainly used for toxicological purposes including determining concentration of drugs in blood and saliva. We can also use it to quantify THC content in marijuana, which is now required by many state and federal laws. One of the cornerstones of a biological unit at a crime lab is DNA sample processing. Our lab is set up to take a DNA sample through the entire analysis process. This process of analyzing a DNA sample occurs in 4 basic steps: extraction, quantification, amplification, and capillary electrophoresis or data analysis. For extraction, we currently use a manual organic DNA extraction where samples are processed in our ductless fume hood we looked at earlier. This process releases the DNA into solution so that it can be further analyzed. The second step of the DNA analysis process is quantitation. We have to determine how much of the DNA we are able to extract because the third step of the process, amplification, works best with a specific target amount of DNA. We currently use the Applied Biosystems Quantifiler Trio Human DNA Quant Kit to perform real-time qPCR. We use the Thermo Fisher Scientific QS5 instrument to do this. This same instrument is found in many forensic DNA laboratories. That third step of the DNA process, amplification, is where we use a series of chemicals and heat to mimic the body's own natural processes for copying DNA to make millions of copies of the specific DNA sequences we're interested in. At the center of the future of forensic science, we use the Applied Biosystems Global Filer Human DNA Amplification Kit. We use the Mini-Amp thermocycler to accomplish this step. The Global Filer Kit is one of the amplification kits used by many forensic laboratories because the profiles it produces are suitable for upload to CODIS, our nation's forensic DNA database maintained by the FBI. In the final step of DNA analysis, we need to visualize the amplified DNA. We do this by running the samples on the 3500. This is a capillary electrophoresis instrument that separates DNA fragments by their size and charge. It then uses a laser and CCD camera to read and record the fluorescence produced from each fragment as it passes the detector window. The fluorescence measurements are then processed with a special software that allows an analyst to determine the DNA profile for that sample. The 3500 is the only instrument of its kind, and it is used exclusively in crime laboratories for DNA profiling. Thank you for taking a look around our lab. If you have any questions or would like to find out more, please don't hesitate to reach out to us at the contact information on the screen.